Okay, אני לא מדבר עברית טוב, so it has to be English. And I'm going to talk about biology. Cells, genes, life, death. Not interesting for everyone, but I think there is a lot of concepts and general themes that you will identify applied to something very challenging, very complex, but also very meaningfully. So I think there's a lot to listen to and to learn from. But first, before I talk about the modeling and the data and the challenges, I want to get you all on the same page, why it's important to model the immune system, what can you gain from it. And you all know Moore's law from compute. In R&D, in pharma, and drug development, there's basically the inverse law, which is called Jerome's law. And it means that every year, the return of investment for drug development is diminishing, meaning you're heading towards a negative zone, where it's just not profitable anymore. And that means that, tacheles, the drug development process is broken. We don't know how to do it well. And there's a couple of reasons for that, but one key factor that is really important is that you're not the same person. You might respond to a certain drug very well, while another person might have a severe tox event or no effect at all. And we know today that in, let's say, oncology, in cancer, in autoimmunity, in neurodegenerative diseases, the immune system plays a core role in mediating the disease and the drug effect. And also the immune system is very, very, very different between people. So the opportunity here is, if you were to understand that complexity of the immune system, not only in one person, but across many, many people, you could decode that complexity and then understand who is more likely to benefit from a drug and who isn't. And this is where the company comes in and what we're building towards. So there's really two pillars. One is creating our own data foundation, meaning heading towards an immune cell atlas of 1 million patients, all mapped at the highest resolution of molecular data and clinical metadata, which then on top fuels the AI that we're building on top to help with the decision making. And what do I mean with decision making? So, like I said, drug development is fairly broken, and there's many decision points in the whole journey. The whole journey from the first identifying a target to actually having it approved by the FDA, for example, on average 15 years. Yeah. Many decisions on the early ones, what target do you engage? What do you want to drug? And then later decisions when you actually are in the clinic, what people should you enroll? What dosage should you give them? What combination therapy do you want to give them? In what cancer indication? So very, very critical decisions that are very costly and usually very ill-informed. And that's how we're coming in, where we're building a platform that really consists of 4 key pillars to help with that decision making. The first one is our own data, like I alluded to, meaning we generate the data in-house, clinical samples, blood, tissue, experimental systems, and creating very, very rich and deep molecular profiling data. All this goes into our central database, which we call Amica, which is basically a cloud-native hosted on top of BigQuery, which then in top fuels the very, very data-hungry models that we're building and training. And the key task of these models is usually, if you want to simplify it, to make good predictions. Yeah. Is this patient going to respond to this drug? Are they going to have a tox event? Would they maybe benefit from another drug instead? So that's the key task of the modeling system, but it doesn't stop here, because in biology, predictions alone are really not enough. You have to explain the predictions. A, to evoke trust, and B, to learn something to develop the new generation of drugs better. So there's a fourth pillar where we use the insane power of the LLMs and their knowledge and their reasoning capabilities that are evolving every day to basically go from these static observations, this patient's going to respond because these cells or genes are important, to translate that into mechanistic hypotheses. Yeah? Why are these genes or cells important? What is the underlying biology that is happening in the body, to then drive the decision-making in a more informed manner. Unfortunately, because of time constraints, I'm going to be talking about the third pillar only, but if the fourth one, the reasoning, the LLMs, is your cup of tea, talk to me, talk to other people at Immunite. There's currently a team lead position opening to really drive this fourth pillar. And I think it's a very challenging and exciting opportunity. Okay, so modeling. But before we go into the depth of it, I want to make sure we all talk the same language, because I know not everyone here is in biology. So when I talk about genes, high school biology, yeah, this is the stretch on the DNA that encodes a protein. Yeah, and protein, think about it, that's the molecule that exerts a function. Then we have cells, I used that word already, which is basically the smallest limbic unit of any organism, or any cellular-based organism. And the cells are capable of performing all essential life processes. The muscle cells, they contract. The neuronal cells, they emit electric pulses, and so on and so forth. Then you have a cell type, which is a category of very, very similar cells that share specific function. So, for example, the skin cells in your body, they're more similar in function and how they look compared to the neuronal cells in your body, for example. And lastly, gene activity or gene expression, that refers to how active a given gene is at any particular moment in time in a cell or a cell type. Yeah? Brain cells express brain genes, muscle cells express muscle genes, for simplicity. Okay, so what's the data? This is a snapshot. This changes every day and obviously growing quite rapidly. Today we're talking about 170 million cells from human and mouse, mostly clinical samples. Talking about 36,000 from about 12,000 patients, meaning that we have multiple measurements per patient. And as you can see here, the emphasis is really on oncology, but we're working in any domain where the immune system plays a role, which is basically in any disease, actually. So for simplicity, what is actually the data that is underlying this pie chart, right? If you, at the bare minimum, in the most essential way to look at it, it's a cell-by-gene matrix, okay? So the human body, the human genome, the human DNA encodes about 20,000 genes, give or take. And for every sample that we profile, we measure about 5,000 immune cells, again, give or take. So every sample, you can think about this dimensional cell-by-gene matrix, where the integer counts here, they represent what I said about the gene activity. How active is this given gene in a particular cell? Yeah? And then, of course, you have many of these matrices, like a tensor, associated with clinical metadata, lab metadata, technical information, which all is linked and connected in the Amica database. Okay, so now how to model this. There's basically a two-step approach that we're using here, deploying the transformer architectures that have worked so wonderfully in many other domains. The first step is really just to go from this cell-by-gene matrix to meaningful and rich cell embeddings. Okay, and I'm going to talk about how we're doing this. The second step is then going from these cell embeddings to meaningful sample or patient representations, because essentially all the questions that we're asking, it's not about is this cell responding or not to a drug, is this cell going to be toxic or not. We want to make statements and conclusions on the patient or the sample level, and here the cells are means to an end for that. Okay, so for the cell embeddings, basically very high level, but I'm going to talk a bit more deeply about what we're doing. So the input, again, is this cell-by-gene matrix that I talked about. But the question is, how do you use this tabular data to feed it through a transformer encoder, right? So there's a pre-processing step, a tokenization step that we're doing. We're not using the raw counts. That's misleading here, the mask counts label. We're using a rank value-based encoding. So what does it mean? For every gene across our hundreds and millions of cells, we normalize the expression to the expression in all the other cells. And the idea here is that you don't care about genes that are stably high expressed or low expressed across all the cells. You care about the genes that are different between cells. Yeah, that's what this normalization step is supposed to emphasize. After that, for every cell, we rank order the genes by this normalized gene expression, and then mapping the gene IDs, TP53, for example, onto token inputs. And then finally, about 2,000 of the top-ranked genes we use as input to feed it through a BERT-style transformer encoder, where the self-supervision training is just masked gene reconstruction, for example, like you know it from language. And after that training step, once the model has converged, you can retrieve the cell embeddings, for example, by the last transformer layer using the CLS token or some simple mean pooling. Okay, so how does it look like in real life when you start with the training? So here, this is a 2D embedding. Every dot is a cell. The coloring here is different cell types of the immune system. And you can see that the model without supervision learns the structure of the cell types. Yeah? So for example, it learns to separate, for example, the killing immune cells from the antibody-producing immune cells. Yeah? And all of this is learning independently from the gene expression patterns. We can quantify that, of course, using very familiar metrics from other domains, mutual information, AUC, balanced accuracy, seeing that all of these improve with the amount of training, depending on how granular you look at the different cell types. But the point here is not necessarily that you can improve on this problem with training. I think the more nuanced or tricky lesson learned here from this plot, if you look at it more carefully, is that it's actually a fairly easy problem. Yeah, so even shallow models or simple models, they do quite well in separating cell types because the signal is so strong. It's the one signal that you pick up immediately. So in order to make good and meaningful or better clinical predictions, the magic happens more when you encode the within cell type variation in the embeddings, and more importantly, how you aggregate these cell embeddings then into the sample or patient level. And this is where the second step comes into play. So here the input is, of course, these cell embeddings that we generated in the first step. And now we represent, what we're doing on these cell embeddings is we're clustering them using k-means or Louvain community detection on a very granular level. So 128, 256, very granular clusters on top of the cell embeddings that in our view represent very, very granular and functional cell states. Yeah, not the T cell, the killing cells or the antibody, but more variation within these broad cell types. And then we represent each sample just by the proportion or by the counts of these cells along these fixed clusters. And then again, for the self-supervision, we do rank value encoding, meaning we mask some of these counts across the samples. But importantly, at this step, we're also adding supervision. Specifically using triplet loss, which I'm going to talk about in the next slide. But essentially, after this training step or pre-training step, you can also get the sample level embeddings again from the last transformer layer. And you see here also, even with very mild or no supervision, you see a lot of structure emerging, samples from the tumor, samples from the blood, from experimental data. They start to align in the latent space. Okay, so triplet loss. This is familiar probably for people, for example, in face recognition. And let's say you have images of people, sometimes from the same person, some taken a day, some at night. And in the end, the model should not necessarily learn the difference between day and night. The model should learn this is a similar or it's an identical face of person A taken a day or night, and this is a different face. Okay? So what you would do here, you would have an anchor. Anchor is person A, image taken on the day, for example, and a positive pair is another picture of person A, for example, taken at day. And a negative pair in that triplet loss example would be another person that should not be similar in terms of the representation. So what the loss essentially optimizes is just the margin between the positive pair to the anchor versus the negative pair compared to the anchor. So what sounds maybe quite straightforward, at least conceptually, when you do this in biology, it's very nuanced, so it's not that trivial. So for example, let's take an intuitive example of you want to separate COVID from healthy blood samples. Intuitively, you would say, okay, I want my COVID anchor, the positive pair be another COVID sample, while the negative pair might be a healthy sample. But when you actually look at the underlying metadata and the variation that is driving the biology, there's many more nuances. It's not only COVID versus healthy, it's also the severity of the COVID, or at what time point was the sample taken? Is it a day during the acute infection? Is it 3 years after COVID? And because we're not in vision or language where everything is intuitive to us, it requires a lot of biological knowledge, good data science, and understanding of the metadata to in the end make meaningful triplet pairs. And in the end, what we consider a positive anchor is a pair that shares all the metadata we care about, whereas the negative pair or negative example differs slightly in one or two of these categories. Okay, so now you want to understand if you're learning something, right, and how you compare to other models, to other solutions. And again, different to the LLM world where there's leaderboards and every week there's a new winner and new records and everything is so standardized and canonized. In our domain, it doesn't exist really. So we have to build the evaluations and the benchmarks ourselves. And we're also building a benchmarking app ourselves in-house where we basically separate the data that the model sees during pre-training from completely held-out benchmarks. And then using these held-out benchmarks and the model inference mode of the trained models to assess, to get the cell embeddings, get the sample embeddings, get the direct predictive power of these models, to evaluate what is the quality, where are they outperforming, what are their weaknesses, and all of this is orchestrated on top of the ClearML pipeline controller. And we really started doing this seriously 2-3 months ago. So today we have 6 models in this warehouse, public models, our own proprietary models, complex one like I'm showcasing here, simple solutions. We're assessing them about in 25 clinical problems, response, non-response, tox, non-tox, before, after treatment. And we're, of course, looking at the data or the outputs of the performance, looking at many metrics to really understand how the models perform or don't perform. And the beauty here is, I think, that right now we're at the stage where every sprint or two, we're adding just more metrics, more nuanced metrics, more clinical problems, more models from the public, more models in-house to really get a holistic view of what these models are doing well or not. So how does it all come together? So this is the one slide I'm allowed to share it in the wild and publicly. So every row here is a clinical problem. Yeah, easy ones. Can you separate a tumor sample from an adjacent normal? Of course, very high performance, very easy problem. To more nuanced, but maybe more interesting problems like can you predict who is going to have long-term survival from a blood sample before treatment? Yeah, very, very, very challenging but meaningful problems where the performance is lower, obviously, because the signal is weaker. And although the model that I've showcased here is in very, very early prototyping, just starting to experiment with architectures, just starting to train with more data, the GPUs, just starting with the supervision, the triplet loss, we can already see that across these very diverse tasks, this model performs similarly to more specialized solutions, which are currently the industry leading models, which are basically more shallow neural networks, variational autoencoders with some linear classifier on top that are trained on every problem specifically, yeah, because they would get confused if you train it on all of them. So very encouragingly seeing on-par performance already, and we really believe that now as we scale the data, as we scale the compute, as we're getting wiser with the architectural decision, that we're soon going to see very strong outperformance or overperformance. Okay, there's a slide missing, but I think the one slide that I put maybe after I send it was how this connects to our roadmap. And this is really a line of what I said, meaning that we're investing heavily into the data, 170 million cells today. By end of 2027, we want to talk about 1 billion cells from nearly 100,000 patients. We want to get more deep with the clinical metadata. We want to look at more modalities, connecting, for example, this foundation model with other existing foundation models that are built in the public for pathology, for clinical metadata, to combine them in the end to get the best of all the worlds, to get a holistic view of how a patient looks like, and then to make decisions. Is this a good patient to enroll in a clinical trial or not? So I hope I gave you a tiny glimpse into the challenges and opportunities that we're facing. And again, if you're bored by language or vision, if you want to jump into very, very new upcoming domains, if you want to work on something meaningful, talk to me, talk to Nir Ratna, who is the mastermind behind the foundation model, Ronny Rechtmann is in the audience. She's driving the data science behind the triplet annotation. So talk to us if you want to work in this domain. Always happy to chat and always happy to discuss. Thank you.